Hey folks, and welcome to UFABC campus. In this class of BECN, we were introduced to the lab environment, and as the final project of the subject, we chose to analyze the sterilizing efficiency of household cleaning products. This is our group and our professor, Professor Schoenmaker. Being present in an environment means being in the presence of microorganisms. Several studies point out that each environment has a diversity of beings that can infect us and cause diseases, and one of the ways to prevent this from happening is to maintain good hygiene, whether local or personal. Many pathogens can survive for a long time on surfaces and can become a source of infection if cleaning and disinfection is not efficient. To reduce surface contamination, it is necessary to maintain a satisfactory and effective cleaning routine. For this reason, the market has several products that can achieve this purpose. Thus, this work has as its main objective to search for products offered in markets that are suitable for cleaning environments, especially homes, and, through sanitizing methods proven by health agencies, verify the efficiency against microorganisms found on surfaces. The objective of this work is to verify the sterilizing efficacy of cleaning products found in general stores. There we prepared a culture medium for a microbiological assay. For that, we prepared 4 petri dishes with 15 milliliters each of medium, containing algae broth, bacteriological agar, and glucose. First, we divided our petri dishes in 3 parts. One with positive control, other with negative control, and the largest one, the test area. The objective of the negative controls was to verify if the petri dishes may have been contaminated during the sample preparation. The positive controls had the objective of verifying if the surface was contaminated with microbiological organisms before the cleaning process. And the product controls, in each dish with a different product, had the objective of verifying the efficiency of the products in cleaning the surface.
The surface chosen for the experiment was the handrail of the first floor of the Alpha 1 building in the São Bernardo do Campo campus of UFABC. The samples collected before the cleansing were collected with autoclaved swabs, swabbing it 3 times on different sides of the swab. The cleaning was performed according to the indications of the government of São Paulo, found in the booklet of Fundação Casa, and in the Manual de Orientações de Limpeza, Higienização e Conservação das Unidades de Saúde, by the Military Police Department of Distrito Federal. These documents were chosen based on the standardization of activities oriented towards environmental hygiene in contaminated places, in order to minimize the risks of transmission of microorganisms, ensuring comfort, hygiene, and well-being to users of such places. The samples collected were swabbed in the petri dish just like we showed before. All the experiments were carried out in laminar flow hood. After the petri dishes were swabbed with the samples, they were stored in the lab stove at 37 degrees Celsius for 4 days. After these 4 days have passed, we got the results. To quantify the proliferation of the cultures, it was applied a grid method to count the percentual amounts of bacterial growth in each section of the petri dishes, and those are the results that we got. We also used a qualitative analysis for the experiment, the cross method. This method is a form of direct quantification, where the greater the number of crosses, the worse is the effectiveness of the product being used, with bigger organism growth. And the smaller the amount of crosses, the better is the effectiveness of the product, with less organism growth. The minus sign, on the other hand, indicates no growth. Comparing the positive control, which corresponds to the surface before sterilization, with the test area, all tested products showed effectiveness in preventing the growth of microorganisms, although it is necessary to note that the growth before sterilizing the surface has been different for each positive control. It is unknown if the handrail was clean prior to the experiment. This is a factor that may interfere in the effectiveness of these products in other surfaces.